There was a lot of work during 2021 to try to help educate people about what this new vaccine means and how it works and why it was done, how it was done so fast. People were nervous that it was done so fast. It could be told as a 1-year story or as a 3-year story of pandemic preparedness or an 8-year story of structure-based vaccine design or 10, 15, 20, 40-year story of HIV vaccine development driving all this technology. It wasn't really a 1-year story. It was a 40-year story.
Welcome to another exciting episode of National Champion Radio, where we celebrate U.S. citizens who have won the world's most prestigious prizes. Today, we welcome Dr. Barney Graham, a man who has quite literally altered the trajectory of human history as the lead architect of the COVID-19 and RSV vaccines.
Because if you can disrupt the function of that protein, the virus can't get into the cell.
Dr. Barney Graham is a world-renowned virologist, the former deputy director of the NIH Vaccine Research Center, and a 2025 inductee into the National Inventors Hall of Fame. He was suddenly thrust into the spotlight when the COVID-19 pandemic broke out onto the world. He rose to the challenge, and as part of Operation Warp Speed, he worked tirelessly under pressure with the help of his colleagues at the NIH and Moderna to bring a vaccine to the world in record time. His work with Dr. Jason McClellan on protein structures provided the foundation for this miraculous, yet also very controversial mRNA vaccine. He now serves as the professor of medicine in the Division of Infectious Diseases at Morehouse School of Medicine, where he continues his work advancing vaccine science and mentoring the next generation of researchers. With that, Dr. Barney Graham, welcome to National Champion Radio. Thank you so much for joining us.
Yeah, thank you for having me.
It's an absolute honor to welcome you to National Champion Radio. So it's really an honor to have you on the show and to have you aboard. You just received the National Inventors Hall of Fame earlier this year. You were inducted, and that's one of the biggest prizes for inventions. And we see your contributions to saving lives directly. I guess take us back. I'm sure you've been here many a times, but take us back to 2019, late 2019, early 2020. Something's happening out in Wuhan, China, and it's spreading and things are happening. You're a man who, creates vaccines and engineers them. And so I guess you were already sort of at Moderna or sort of in that space working on something like this. But how did everything happen and what was that like? That must have been quite the whirlwind, adventure, et cetera.
Yes, 2020 was a banner year. But I started this path a long time ago. It really started when I ended up at Vanderbilt for my medical training, post-medical school.
Okay.
And saw the first AIDS patient in Tennessee back in 1982. And that's what led me...
Oh, the first one?
The first one in Tennessee, right.
Oh, I see.
In the fall of 1982. It had been reported a year before in Los Angeles, and it was because of our work on HIV vaccine evaluation and my work on another virus called respiratory syncytial virus, RSV, that more people are starting to hear about.
Yeah.
That work in the lab on RSV and the work in the clinic on HIV gave me the opportunity to be one of the founding members of the Vaccine Research Center at NIH in Bethesda, Maryland. It's part of NIAID, part of Tony Fauci's old institute, but it's on campus. It's an intramural program, meaning that 90% of NIH money goes out to the universities, but some of it stays on campus. So I was a federal employee starting in 2000 when the VRC started. And that work that was focused on trying to make an HIV vaccine over those 20 years really led to a lot of the technologies that we used, first to solve the RSV vaccine problem. And that was done by finding a way to hold the main protein target of RSV for a vaccine in a better conformation, a better shape, a more native shape, the functional shape of the protein. And solving that, using crystallography and solving the atomic level structure of that protein and getting it stabilized in that native conformation, all of a sudden gave us a protein that could be a vaccine. And that happened in 2012. And so now, 10 years later, that protein has led to vaccines for RSV made by Pfizer, made by GSK, and made by Moderna. So there's 3 vaccines approved for human use, either in the elderly or maternal immunization for RSV.
Wow.
And that idea that if you got a protein in the right shape, if you got it in the better shape, the native original shape before it changed and rearranged itself, that it would make a better vaccine, we tried to extend that work to other viruses, envelope viruses. And one of those was coronavirus. And so we wanted to extend our work on RSV to another envelope virus family. And so we chose the coronaviruses and worked on one of the original coronaviruses called HKU1, and also on the MERS coronavirus, which had broken out around that same time we solved RSV in 2012 and 13. That was the coronavirus in the Middle East.
Yeah.
In Saudi Arabia. And so by 2016, working with Jason McClellan, who was then at Dartmouth University up in Hanover, we were able to eventually get the spike structure of a coronavirus, a human coronavirus, working with Jason McClellan there at Dartmouth and also Andrew Ward at Scripps. So the technology is fabulous, and it helps you really know and map exactly where you want an antibody to land in order to disrupt the function of that protein. Because if you can disrupt the function of that protein, the virus can't get into the cell. And so in 2016, we got the spike structure of a human coronavirus called HKU1. And then we learned how to stabilize it. And we found out that if you did that, the spike on a MERS coronavirus would be a really good vaccine antigen. And because of some other work that we had been doing on other outbreaks like Ebola and Zika, by 2016, we had also learned that mRNA had become a good delivery mechanism, better than the DNA that we had been using. So there's more than 100 different virus families in the world and on Earth, but only 26 of them are known to infect humans. So the idea was if we could limit our work to those 26 virus families and find a prototype within each family that we could work on in great detail to find solutions. So we made a deal with Moderna to use mRNA, which is a rapid platform type of manufacturing. It can go faster just because it's synthetic, and you do it the same way for every product. And we did a big project on the coronaviruses family and the paramyxovirus families. And so we had a prototype, the MERS coronavirus for coronaviruses and the Nipah virus for paramyxoviruses. And we made vaccines that we thought were generalizable to other viruses, and both by RNA and by protein. By the 2019, we'd already shown with Moderna that we could protect animals from lethal challenge with those pathogens, with those viruses. And so this was our project. This was more like a demonstration project that we had been working on for years.
Yeah.
And so, and in fact, we'd already paid Moderna enough to make us a batch of RNA to test our Nipah vaccine in humans in the first quarter of 2020. We'd already set that up in September of 2019.
And so this is before the outbreak. You guys
This
paid
was
them.
before the outbreak. And then when we heard about the new virus at the end of 2019. And then on the 6th of January, we heard that it could have been a beta coronavirus. That means that we had a solution for how to make a coronavirus vaccine. So we said, let's hold on the Nipah virus product and let's make a vaccine for this new virus as a demonstration project. And so when the sequences came out on the night of the 10th of January, those sequences were posted online by an Australian and Chinese investigators that had done some sequencing. And so we learned the sequence of the spike protein, meaning the nucleotide sequence, the basic genetic basis for how cells make proteins. So we
Yeah.
learned that sequence.
That's the whole ACTG
And
whole thing. Is that right?
right, that's the ACTG alphabet
Yeah.
for making codons that then put the amino acids
So
in the
you
right
you had the
position.
entire sequence of this new coronavirus in your
Right,
hands that got published, globally because there's
right,
great, global shared information, right?
right, exactly. And so we could align our known sequences of coronaviruses with this new one, and we would know where to put our stabilizing mutations into the new protein. And we could just make that without any additional experimentation because of the work we'd already done. And so we did that as a demonstration project, really, because this was before the virus had come to the US. It was before it was really thought to be a pandemic problem.
Like a global
And so,
pandemic.
right. And so we made all the reagents. We ordered everything we needed on the 11th, as soon as we got the sequences, and started making proteins so we could solve that structure, and started making assays so we could measure immune responses. And we sent a sequence to Moderna and asked them to make us an RNA for that sequence. And then around the 4th of February, we got it back from them. And at that same day, we had mice ready to immunize. So by
Wow.
February 18th, we knew that our new mRNA construct was very immunogenic, meaning it elicited a very good immune response. So, so we had a lot figured out. By the 1st of February, we had the atomic level structure of the new spike protein. We had ways of measuring antibodies. By the end of July, we were able to start a phase 3 trial because we had animal data for immunogenicity by starting the phase 1, and now we had at least some safety data before starting the phase 3. And about 200,000 people were enrolled over those next 3 months in order to start finding out, does the vaccine work? And so by the 9th of November, I think that's the day I found out, Sunday, that the vaccine indeed did work. It was working at a 94%, 94.5% protection rate, even after the first dose.
Wow.
And so it was a very remarkable year for people like me who spent 40 years in the background just trying to figure things out. It's that kind of work is really a grind. It's not a big thing that you see in the newspapers or on the
Yeah.
news. It is a daily grind. Every now and then you find really interesting things, but most of it is not newsworthy.
Yeah.
And so it was a interesting year to be involved in something that really started out as a demonstration project, but then as the next few weeks went by, the stakes got higher, and it was
Yeah.
important that it then worked, and we were very fortunate that it did work. That our guesses at the right sequences turned out to be the sequences. In fact, those sequences that we selected were used in the proteins for not just Moderna's vaccine, but for the BioNTech vaccine that Pfizer got, for the J&J adenovector vaccine, for the Novavax vaccine. all the vaccines approved for use in the U.S. had that same protein sequence in them.
Wow. Okay. Wow. And so, how did you guys eventually figure out that that was that right protein sequence? You got it in January. You saw where the holes were at, I suppose, in the sequence, or where you could put in, some of the tools you had built into that sequence, and you kind of
Right.
knew, or at least had a guess, a hopeful guess, Hail Mary, maybe. I don't know how on the scale of certainty you were at about it, and then eventually have to push it out prior to knowing at scale, because you can only do a certain amount of trials before we're at a time where we have to just take the chances because we have a global pandemic on our hands and the
Right.
numbers aren't looking great, and the walls are closing in, and everybody's getting literally closing in inside, and it was just a crazy time, and here you are at the forefront daily having to wake up and I guess hustle towards trying to find solutions, right? So I guess
Some of
within that
those
beginning,
choices,
yeah.
we made were based on our prior work, because in 2017 we published a way of stabilizing the spike to make it that better protein, and we had shown that that same approach had worked in about a dozen other coronaviruses. So we were pretty confident that if we aligned those sequences from the new one and the old one, and then we put in two prolines between two places that have to pivot in order for the protein to move. And the
Okay, so this is like you putting
prolines,
a. Oh, go ahead. Sorry.
so the prolines are an amino acid, one of the 20 amino acids we use to build our proteins. We knew that we were going to put the prolines at position 986 and 987. And that had worked before. In this case, since it was a demonstration project for us initially, we said, let's go with our prototype approach and see how it goes. Typically, you would make 100 different iterations of that to see which one would work the best.
Maybe you try
Yeah.
95 and try 496 and put a
Right.
kink at.
Yeah.
I
And
don't know. I don't know if these are any terms that
right.
make
And
any
so
sense,
there's
but
there's a number of approaches to stabilize these proteins. And so
I see.
to to just select one and go with it without any additional experimentation was kind of a big risk in a way. But we were pretty confident. If we had known it was going to be that kind of a pandemic, I don't know if we would have had enough courage to have done that. But since it was a demonstration project, we had enough courage to go ahead with it, and it
I
turned
mean, what
out.
does that really mean? I guess. I mean, I get that it's not the real thing, but I guess it means that you're going to go take that approach that you created, present it to someone, and say, "Look at what we've done with this. Maybe give us some more money to go back and try 10 other approaches, or based on what we've seen from this approach, we might try it in animals, and then go back." I don't know. Yeah, what does that
Right.
really mean at that level?
We were trying to promote an idea and a concept for how to fund basic research on viral families that would give us the information we needed to make a rapid response if we had to something else. And so we wanted to just show that if we had to, we could make a good vaccine within a few months. And so if we could just show how fast we can make a vaccine, if we had this prior information, then we could, use that idea and way of thinking and then way of organizing the research we needed ahead of time to be better prepared for pandemics in general. So that was really what we were working toward. But as the weeks went on, we realized the stakes were pretty high and
Yeah,
that
I'll
it
say.
really needed to work.
Yeah.
And fortunately, it was. I mean, you can make it slightly better with a few other modifications, but the version we chose was quite good, and it worked. It worked for the most part as well as it could have.
Yeah, wow. And I'm envisioning. Tell me if this is maybe a proper bit. You got the two versions of these proteins in the different shapes that they take, right? And so they fold and flip, and they do all types of iterations. That's how proteins work, and that's what makes them different proteins from one another. Their functionalities are based in that shape. And we saw Google's big protein folding, discovery come out some time ago. I don't know how many years that was now. And that was DeepMind. And so essentially people were trying to solve something similar in coronaviruses by trying to essentially stop the function of the later state of this protein. And your part of your innovation was replicating firstly, and then being able to stop the folding of the earlier stages of this protein. So you say, okay, we got this shape. Now it wants to try to bend like this, and we're gonna put something in the way, like a axle in a wheel or something like that, like a stick inside of a bicycle or something. We're gonna stop it from doing the folding that it wants to do at this point, and that's gonna be enough to stop its entire functionality. If I put two stakes in it here, it can't flip. And then if it can't flip, then it can't bind to the next one. And all of a sudden, we've broken this entire chain that happens with these proteins.
Right. And you want to hold it in that original shape, because the way the vaccine works is you show the immune system this protein that has all sorts of shapes and contours in the surface. And, antibodies have to fit into those little spaces just right in order to work. So you give the body this protein so the antibodies can be selected that can recognize that protein in its original shape. So if you can generate those antibodies and make the B cells expand and then have antibodies on hand ahead of time, that's what gives you the immunity that would allow you to respond to the virus a day or two faster than you would have otherwise. And that makes all the difference. If your immune response can get there 2 days earlier than it would have, it's the difference between getting really sick and being asymptomatic. So the whole idea here is to get your immune system primed so that it can respond quicker to the infection. And it can drop down the amount of virus that you're infected with before it generates all these immune responses and causes all this tissue damage. So that changes it from a mild, short-term infection to instead of something that causes lethal disease.
Wow. I just had a vision of like a twin almost that you're creating ahead of time to show like a criminal, like this is what you need to look out for in the criminal, and essentially you get all these people prepared for the next time they see that criminal or the same exact thing.
Exactly.
But it's hard to make a twin, right?
It's like facial recognition. It would
there you
be
go.
like that. It would be just like you're saying. It would be facial recognition of somebody that you really have to look out for.
Yeah. So here's a dangerous criminal. It's Barney Graham. He's armed and dangerous. You got to watch out. Here's what he looks like. And then, a week, so everybody's getting prepared for it. He's going to come to Detroit, and this is exactly how he's probably going to try to attack us. We're going to get the exact responses ready. He's not an airborne kind of guy. He's somebody who likes to drive on the freeway fast and whatever the antibodies are for that.
As soon as he crosses that bridge, you're going to see him.
Right. And you have to have the right antibodies, which is the right kind of defense for exactly the kind of attack that Barney Graham is going to bring, right? So it's like, we need the Air Force in this one, or we need SWAT, or we need whatever is best for him,
Yeah, exactly. You have to show the right image to have the right response. Exactly.
Wow. Okay. And so, I mean, maybe we go back and compare it to what's so difficult about AIDS and HIV and AIDS, and also that time, I guess. I mean, I'm curious because I was thinking when you were saying it earlier that like maybe a lot of young people. I mean, I wasn't born when AIDS happened, right? I was born in the 90s. It was still sort of around. I mean, obviously it's still around, but you know what I mean. The big outbreak I think was late 80s, mid to late 80s, right? And so that was a really scary time for people, right? I mean, it wasn't
between 1982 when I saw that case and 1996 when we finally had drug combinations that could control the disease. Everybody died.
My
Everybody
goodness.
who got infected died. And those 14 years were really, really hard.
Yeah, I can only imagine. I mean, it's interesting how how that time must have been like, and a similar space of trying to find cures, and it made such a big impression on you that it turned into your life's work. And I think bringing people back to that time, younger people, having them experience, think about what that was like, and also how it compared to the pandemic that we all did, or most of us did live through, right? And so, yeah, I guess I guess one big way you comparing it is that it was a lot longer. It was a lot more lethal once you got it. Maybe it didn't spread as easily, but once it got to someone, that was it. There was no going to the hospital. There was no chance of, I mean, you tried and you fought back, but in the end, it just killed everybody.
Right. So
Yeah.
in coronavirus, the SARS coronavirus, almost half of the people who get infected don't really have symptoms. They don't even know. So they are innately primed in a way that they can respond to a virus and help clear it gently or early so
Mm-hmm.
that it doesn't cause a lot of disease. But about 10 or 20% of people got pretty sick and had to be hospitalized, and about 1 or 2% of people were dying. And so, you know, it is complicated when a lot of people get infected and don't really get sick,
Right.
because then they think it's not that bad. It's not that bad of a problem.
Those secret
But when you
spreaders.
start adding up, when you start taking 1 or 2% of a large population, that turns out to be a lot of people. And 10 or 20% of hospitalization was enough to basically shut down our whole healthcare system. It overwhelmed our healthcare system. You know, a little over 1 million people died in the U.S. from coronavirus during that year and a half, 2 years.
Wow.
So it was devastating, even though some people didn't get that sick. Now, the fact that some people can control the virus and clear the virus without getting too sick, that's a good sign for being able to make a vaccine,
Really?
because you know that immunity works. For HIV, when nobody cured, when nobody's self-cured, when nobody's self-cleared, when you can't recover from a virus infection naturally, it makes it a lot harder to think how to make a vaccine that could be better than natural immunity.
Hmm.
And so HIV has a lot of problems. I don't know if you have time to get into all the issues with HIV that make it a more difficult vaccine target, but the
I have
one
all the
thing,
time. I don't know if it's up to you
yeah,
if you I'm here for you. I'm very we have all the time. But
but the so going through the reasons why we don't have an HIV vaccine would take some time, but the act of trying to make an HIV vaccine over these last 40 something years has led to the technologies that allowed us to make the RSV vaccine and the coronavirus vaccine. And so, all the tools we needed for what we call structure-based vaccine design, and all the tools we needed for surveillance that, you use to both discover the genome of the new virus or track how far it's spread or track how prevalent it is in a population. All the amazing immune technologies that we can use to measure immune responses now, a lot of that was all because of trying to make an HIV vaccine. So even though we don't have that vaccine, that work to try to make an HIV vaccine has had a lot of value.
yeah, I mean at least I mean to me that feels like a way of saying that all those people didn't die in vain. You know, in some sense the things they went through is open up the doors for humanity to evolve technologically in that direction specifically to then save lives down the line, to save
Well,
lives
we
from
need to,
RSV,
right.
Corona, etc.
That's one way of honoring their memory, is to
Yeah.
give them some credit for all this new technology.
Yeah, that makes that's I mean, and so I mean seeing the human body, seeing knowing so many in such detail of not just the human body but all these other, organisms and non-organ, debated organisms like viruses. What do you make of a human being? Like what is a human being to you? Do you like tie that with like theological ideas, and how do you separate the two if you do? Like where do you land in seeing all of that?
Well, life is a miraculous thing. I mean, if you really think about what it means to be alive, and you think about how our bodies work, you think about where your thoughts come from. Where do all these thoughts come from? You
Yeah,
know, people have thoughts running through their head all the time. Not all of them come out, but
thank
people have
goodness.
a lot of thoughts. Where do all those thoughts come from? And and so for me, who has spent his whole life in biology and trying to understand how biology works, and I see so much beauty in biology. It, not only visual beauty, but just the patterns and the way things work and the way things are regulated and the way things are balanced. And, everything has a purpose and has a counter balance to make it not. So most good things, if you take it too far, will become a bad thing. So everything has a push and a pull.
Yeah,
And to see that in action, and to just watch your grandchildren grow up, it is a miraculous thing. It's an amazing thing. And
yeah.
if young people could see biology the way I see it, I keep wondering why 80 or 90% of our children don't want to be viral immunologists. I mean, why would they want to do anything else? It, it, it just it's an amazing thing to me.
Does
That
it
that's
make
how
sense?
it's how amazing biology is. And so I had a chance this morning to talk to my grandson, one of my grandsons, who has just started at Kennesaw State. He's a freshman, finished his
Oh,
first
okay.
semester at Kennesaw State, and he wants to go into biology. So he's at least picked up some of that. None of my children
He at least
became
gets it.
biologists,
He gets it. He sees it.
but yeah, but one of my grandchildren at least is interested in biology.
Yeah. I mean, I think like that's part of what I'm trying to do here is to distill, the nature of how people get to see the world through the work they do. And just how awesome it is in all these various forms to be human and to see the world through human eyes. And so I think and everybody has their own vision, like you said. If people could see it through your eyes, I can only imagine the intricacies with which you get to see the eventual functionality, the basic functionality of human beings going about their day, moving around. Like you understand how the organs are functioning and the different functions at, the protein levels and what happens obviously when viruses come to play and just growth. I mean, what is it like how does this toddler become this adult, and all these functionalities are like locked in this sort of like, it reminds me of those Russian dolls, because it's like you need human beings to keep making them happen, even though they're birthed like eggs almost and they happen at scale. So it's like, I don't know, it took evolution or whatever X amount of thousands, millions of years to develop a human. And now to get a new one, you just make a baby and it's all the DNA is stored and the information is given. And all of a sudden this unfolding process just knows how to happen. What? Wait, what?
Every one of our cells in our body has the same genetic information,
Okay.
but one cell makes muscle tissue, and one cell makes an eye, and you know every cell has the same genetic information.
Wow.
It's just used and regulated in different ways, and so even the positioning in space will make you do something else even though you have the same exact genetics. That cell will do this thing instead of that thing.
What do you mean, like the way I don't know, like what ends up being the phenotypes of the different cells? Essentially, is that what you're going with?
Right. So, your skin cell has the same genetic material as your liver cells. I mean, all your cells have the same genome.
Right, right,
It's
right.
just a matter of what are the systems and triggers that make that cell make these proteins to do this function versus these proteins to do some other function versus make these proteins to create a different shape of the cell to line your
Wow.
airways instead of make a spleen. All all those cells have the same exact blueprint. They just are using that blueprint in different ways to make different things.
Reminds me of
And
the
that
Yamanaka
just
factors.
blows your mind. And I think we underestimate our children because I know that second, third, fourth graders, if you talk to them about biology and how it works, they get very excited and they have all sorts of ideas and ways of seeing this. And I think we wait way too long to teach our children about biochemistry and biology and things that we actually need their imagination to take some of the next steps. And, we underestimate them. We wait till they're in their late teens before we really start getting into anything that's very substantive.
Yeah.
And I just think we're making a big mistake on our children about how long we wait to teach them certain things.
Wow, that's a big claim. I agree. I mean, and then by the time we teach them substantive things, we got to deal with puberty and we got to deal with all types of other stuff happening with them.
Right.
When early on, they're like there's less distractions and they can process so much, and obviously they can learn the complexities of language and pick up on it. And I agree with you in the sense that having an open canvas gives them a lot more ability to understand and to make deeper connections.
Yeah, I really enjoy being in a room full of 8 or 9 year old children because their minds are just like lightning.
Yeah, yeah. Now I was gonna say you say that, and it's interesting because what we haven't touched on is that you're a professor at Morehouse.
Yes, I am. I'm now, after I left NIH, we had planned to leave NIH in 2020. We made a 2020 plan in 2015,
Oh no, that was
but
2020 had other plans for you, huh?
I had to hang in there till the end of 2021
Thank you
so
so
that
much for yeah.
we had this thing wrapped up. And so we moved to Atlanta. And in 2022, I joined the faculty at Morehouse School of Medicine.
How has that been? I mean, I guess it's pretty recent, but yeah, what made
Well,
you pick Morehouse?
I love
Historically black
Morehouse.
college, and
Yeah, it's one of the four historical black medical schools in the country, along with Howard and Meharry and Drew. It's just gone through its 50th year, so this is the 2025 is the 50-year anniversary of the beginning of the Morehouse School of Medicine,
wow,
and and it's graduated now I think 40 classes.
mm,
So I love being at Morehouse. I came to Morehouse because Morehouse is very devoted to health equity and health justice. And after I saw how our technology was so powerful and so successful, the scientific part or the technical part of making these vaccines was so successful, but most of the good the vaccines did happened at the end of 2021. So in the fall of 2021, when the Delta wave went through the world, that's when most lives were saved. Now, people's lives are still being saved by the vaccine, and there's a big difference if you're vaccinated or not in terms of how sick you get. But most of the deaths averted were in the fall of 2021. By the end of 2021, only 5% of the African continent had been vaccinated. And you got to ask the question why. We made 14 billion doses of mRNA between Moderna and Pfizer and other groups. We had billions and billions of doses. Why did it take so long to get to the low-income countries for something that, for a virus that doesn't discriminate between place or person? And so I wanted to work with people at Morehouse who are experts in articulating the importance of health equity and health justice. So that's what I don't have a lab anymore. And so I'm just, honestly, most of what I think I do is look for talented students and see if I can open some doors for them. That, because there's a lot of talent that I think has not been leveraged, a lot of talent that has not had access to become what they could become. And so that's how I spend my time now.
That's really noble and exciting. And I think, a lot of people say that that's really gratifying to be able to do that, to be able to open up doors and pay it forward. And it's so, I mean, you talk about the human body and then you go up into human institutions and systems as forces of nature as well, right? And meritocracy is so important. Diversity is important for survival in biological systems, habitats, et cetera. And finding or getting the right leaders to the top and not having it be just nepotism or other forces that are skewing the value of our institutions. We need world-class institutions to function as good as they can. Lord knows we need in America. I mean, we see some of the outcomes we're getting, whether it be our K through 12 system or just education in general, or if people have trust in the CDC, or people have trust in and faith in these different institutions, or who rises to power and how, or if this one talented person would have got the right perch from Dr. Graham early on, all of a sudden, 10 years later, they become the right leader for this other thing where we really need a strong institution or strong government or et cetera, et cetera, et cetera, right? And I think these institutions end up shaping and deciding the outcomes in large parts of our biological bodies, right? And so it's so important to me that those systems are treated properly, right? And that they're healthy as well as the human bodies inside of the nation.
So this person I mentioned, Jason McClellan, he was a
Yeah, he won as
postdoctoral
well.
fellow. He was a postdoctoral fellow in Peter Kwong's lab when we started working together on this RSV project back 10 years ago, or 15 years ago now. But and he was a very young person. He didn't know that much about RSV, but he knew a lot about structural biology. And so coming together, I knew where to take it, and he knew how to do it, and we were able to do a really new thing and get a new thing done that has changed, health for babies from now on. But
Wow.
and then he was a big part of the coronavirus work too, and then Dr. Corbett, Kizzmekia Corbett, who's now Kizzmekia Corbett Hilaire. I met her when she was in high school.
Wow.
And she worked in my lab for a few years, including some of the summers during college, before she went to UNC to get her doctoral degree, her PhD. Then she came back to my lab at NIH in 2014 to do her postdoctoral work. And so Kizzy, Dr. Corbett, was there with us when we were doing our coronavirus work in 2014 through 16, 17, stabilizing the protein, doing those experiments in mice to show that the MERS coronavirus idea was going to work with Moderna. And Kizzmekia, she was in the lab in 2020. And she managed 3 other African American students in my lab to do most of the lab work, most of all the data we needed to start that first phase 1 trial in March of 2020. that was because Kizzmekia was there and helping to make all that happen. So Kizzmekia is somebody who needed that kind of visibility. So when President Trump came to visit our lab in March of 2020 to see what we were doing, because he'd heard that we were working on a vaccine, I had her in the room with me. And she got to make some of the presentations at that time, and that's when CNN and MSNBC and all these other places found out about Dr. Corbett. And so she became very well known during the coronavirus outbreak, and she now has her own laboratory at Harvard. She runs her own lab at the T.H. Chan School of Public Health at Harvard. Before the pandemic, we were trying to find a faculty position for her, and it wasn't going that well. I mean, we were finding a few opportunities, but not many. But after the pandemic, after she became so well known, after she became a face on TV that people could see, there were universities out there bidding for her. I mean, they were competing for her.
Yeah.
But she was a good scientist before the pandemic.
She was.
And she was a good scientist after the pandemic. But, it took that notoriety to get over this activation threshold that people need to let Dr. Corbett fulfill her destiny.
Wow.
And so, that kind of, there's people like that everywhere. And that is what I want to spend my time working on
Is this
to make
accurate here?
sure that is the March of 2020. Yeah.
So that's
Dr.
Dr.
Corbett's
Corbett
on
there.
the right. I'm
Yeah.
there. You see me on the left. And
Yeah.
that's Dr. Fauci, Dr. Francis Collins, and others in the background while we're explaining to President Trump what we're trying to do.
And this was in March.
That was in March of 2020.
Wow. And what was your take on President Trump? I mean, how was that at the time? Obviously, things have come a long way, and that's been a whole historical chapter. His first term out of office and then back again now. What was your take on him then? I mean, how does he leave a mind like yours and then go say some of the things he said later about bleach or all these things that were taken out of context, perhaps, or maybe, yeah. What was your take?
Well, we were very glad that he was interested in understanding and learning more about vaccine approaches, possible approaches that could, work and could benefit. This was very early days, and I think to his credit, he let that $12 billion go into helping those pharmaceutical companies figure out how to scale up the products. So before we knew that they worked, there was a lot of work going on to see if they do work, can we scale them? Can we make 1 billion doses instead of a few thousand doses? And so that $12 billion that was spent early on to help those companies scale up is why the vaccine got launched the way it did. So
Yeah.
I
They
think
always give him credit for the task force and for the speed with which he decided to, put that money in the right place and let that process go forward. Of course, he takes a lot of credit for it, and I think people give him credit even if he doesn't take it.
so. I think letting that process go forward, was a big thing during that year. And the Operation Warp Speed actually has a different story, but that was a derivative of what happened during Zika as well.
Okay.
Not only did we learn about mRNA during Zika, but during Zika, the government across agencies got better organized than we were in 2014 with Ebola. And we formed something called the Zika Leadership Group, which brought all the U.S. government agencies involved in this together every couple of weeks to make sure we were coordinated and on the right path. And John Mascola, who was head of the director of the VRC at that time. I was deputy director. He was director. He's the one who got that organized. He's the one who put that Zika leadership group together. And when coronavirus broke out, by March, John had also is the one who organized what ended up becoming Operation Warp Speed. Somebody else named that, but John is the one who started that group to be organized and how to coordinate across different agencies like BARDA and the FDA and the CDC and all the different groups that had to be functional in order to get this big project to happen.
Yeah, I guess going back to the design and those days, I mean, having the president come to your lab, if signs weren't sure then that the stakes were high, right? I mean,
That's
that
when the
was a,
stakes were really high. Yeah.
I
That was the
mean,
beginning of March, early March.
what was it like for you personally? I mean, you have a family, you have kids, grandkids. You've been sort of not in the limelight much for the work, even though you essentially solved RSV already, and so I mean that was massive. That's massive already, and you got some awards for that, I believe, right? And but you weren't really in the limelight in that same way. And the stakes perhaps were never as high with you operating at the highest of your abilities. What was it like in March, April, May, trying to find the right connections, getting up every day? Were you going to sleep stressed? Could you sleep at night, or or was there some balance that you found, in those days? People are dying every day. The news is getting crazy. People having political differences in the world. It was just a time for everybody, really. But I imagine for you, I just can't imagine it. I don't know. I don't know what it was like for you at the height of this, trying to once again solve that jigsaw puzzle of identifying the right thing, and now you've taken your test and you've gotten it out there, but now is it going to work, et cetera? So
Yes, well, it was a year that like no other. And,
yeah.
I spent a lot of my time just trying to make sure my lab could keep working, that they had the supplies they needed, they had the space open that they needed, that they stayed safe, trying to get in to work in the lab.
Wow.
And so, trying to manage young people, coming together to work during the beginning of that pandemic, that was a stressful, difficult time. And we were all working 24/7. I lost 20 pounds during that year
Oh,
just
wow.
because, you're just kind of in it all the time.
Yeah.
And so, and there's 1,000 things that have to go right for a vaccine to work. And usually the vaccines you make do not work. I mean, it's not that common for a vaccine to end up working. And so we had to make a lot of choices, a lot of decisions, and a lot of things had to come out the right way in order for this whole thing to have happened. And so, a lot of things have to happen in the right order. If you make choices or do things out of turn in the wrong order, you can lose months or,
Really?
of
What do you mean
time.
by that? What's an example? Like,
Well, so, you know, we ordered our reagents the day we got the sequences. We didn't wait 3 days to decide how to order.
mm-hmm.
We immunized mice the day we got the mRNA back from Moderna. We didn't wait till the next week to immunize the mice. Those mice had to be ordered ahead of time, and everything had to be in place, and we had to have approvals and all sorts of things in place in order to even start that experiment. We had that initial immune response just in time to justify to launch that phase 1 trial. We had the safety data just in time to launch the phase 3 trial. And, there's a million documents and things you have to line up for the FDA, first to even start the first trial, but then to generate the data and all the justifications and that you need for them to make a decision to say it's okay to go forward to the next stage. So there's a lot of things that have to be lined up in just the right order without wasted time in between.
Yeah.
If you're gonna, if you're gonna get something to happen fast. So,
Yeah.
making sure all those things happened all the way through that system, all the way through to approval, that was just took a lot of focus.
I bet. Yeah. And while you guys are in stage 3, are you already thinking about scaling manufacturing? Is that what you were saying
Well,
that you
see,
guys?
they at the time we were trying to get through all those studies. Moderna's people were figuring out how to scale this up, as were Pfizer and BioNTech figuring out how to scale it up. Because up to that point, people had only made a few thousand doses of mRNA at a time. They had not made millions, let alone billions of doses. And
Mm.
so getting it from a few thousand up to a billion, that is not trivial. Especially, we didn't have enough time to really get the cold chain requirements to the point we'd like to.
What's that?
Eventually, mRNA will probably be lyophilized on a microneedle, and you can store it at room temperature. But during that year of distribution, we had to store the mRNA at minus 80 degrees centigrade.
Wow.
So, there was a lot of work that had to go into keeping cold chain storage conditions right. Most of the time, vaccines that are out there licensed are either at 4 degrees, which is refrigerator temperature, or at the most minus 20, which is a normal kitchen freezer. But having it at minus 80 requires a lot of specialized equipment and dry ice and all sorts.
Whew.
So
Yeah.
there was a lot of things to figure out and organize during that year.
Wow. Yeah. I mean, that's just one thing after another, right? One battle and I guess you just keep your head down and just keep it moving. And,
Right. And then
you know,
the supply chains for the nucleotides that had to be used to synthesize the RNA and all the lipids you needed to package the RNA. And, there was a million things to figure out that year that somehow all came together for us to get it launched by the end of the year.
yeah.
I think the first dose, we just passed the 5-year anniversary of the first injection, first vaccination in the U.S., which is December 14th.
Oh, wow. Oh, wow. That was,
Yes,
yeah, a
so
few days ago.
December 14th was the first immunization in the U.S.
Wow. Well, congratulations on, I guess, reaching that milestone. I
Yeah,
mean, congratulations to all of us. I mean, I think your contributions saved so many lives, like you said, especially that 2021, because then it got hot out and it was a little different, but then that wave was going to come in the winter, and to not have any protection was going to be massive,
yeah,
and it was just really miraculous. Obviously, that miraculous nature also led to skepticism. You're at Morehouse, the head of ethics and rationality, morality, etc. I guess what have you learned from there, or what's the thinking there on how to start talking more broadly about vaccines and acceptance of vaccines, not having trust in government, I guess, is a big part of that. So, I mean, what have you seen, or what's your thoughts on how to messaging, I guess, or the communication gap, or etc.
Well, one of the things that you have to do is communicate. National Heart, Lung, Blood Institute, which is another institute at NIH, was headed by Gary Gibbons. Dr. Gibbons is a cardiologist, researcher person who had been at Morehouse in the past. He had been a grant-funded researcher at Morehouse before he ended up as the director of the National Heart, Lung, and Blood Institute. Their institute developed a concept for grants that would help educate communities.
Mm-hmm.
So Morehouse School of Medicine, actually, before I ever went there, had some of these grants to do community education and vaccine information. So Morehouse was already fully into that before I got there. It's one of the ways that they work on health equity is by doing a lot of community engagement and helping community members have, health leadership within them to help, people make better decisions about their overall health. So
Yeah.
it was a lot of community engagement during those years. During 2021, as the vaccines were coming out, Dr. Corbett and I were on webinars almost every night for months and talked to a lot of groups, sometimes smaller, sometimes larger groups. But there was a lot of work during 2021 to try to help educate people about what this new vaccine means and how it works and why it was done, how it was done so fast. People were nervous that it was done so fast, but
Yeah.
there was probably more data and more, more consideration, more everything for that vaccine than almost any other vaccine I'm aware of in history. I mean, it was a huge amount of data in a very short amount of time, done in a very precise way. So, and like I was saying, it could be told as a 1-year story or as a 3-year story of pandemic preparedness or an 8-year story of structure-based vaccine design or 10, 15, 20, 40-year story of HIV vaccine development driving all this technology. So
Right. Right.
it wasn't really a 1-year story. It was a 40-year story.
Yeah, that makes a lot of sense. I mean, the information, the developments over time, things that happen after Zika, like all these different, bringing these institutions together, a little more structure there led to the, rapid development later, right? The rapid response
Right,
team. So
right.
whether it's at the scientific level of the virus and the vaccines themselves or at the higher institutional levels of organization, a lot of things had to fall in place for those to happen. And
Right.
I guess it's, I don't know, of those worlds, maybe it's like, I guess there's a world where you don't have the vaccine and you don't have that problem of telling people to take it that fast, but then you have the problem of the
Of
virus,
people dying.
of people
Right.
dying, and it's like, yeah, I don't know. I mean, thank goodness you got the vaccine and everything and everybody else who was involved in that chain, things worked out the way that they did. And so with what's happening now, I guess, thinking about the forefront, thinking about the future, thinking about kids and AI is massive right now. Has AI been a tool that's been helping you sort of identify the facial recognition of different viruses as it played a part now? I'm sure some
You
part
know,
of its
the
wizardry is helping out with that.
the group that did AlphaFold, including David Baker at the Institute for Protein Design in
Yeah.
Seattle, who did some of the AlphaFold and other, computational work that was needed to understand protein structure. They got the Nobel Prize, I think, in 2024. So, AI, these kind of capacity to do computational biology is changing a lot of things. It's changing everything. So AI is going to play a big role in the future. AI is not always going to get it right. And AI, which is based on everything we already know and putting it back together, may not be able to innovate. I mean, it may be better to prompt AI to help it prompt you. And then you can be the one who, with your human mind, innovate. I don't know if AI will be able to innovate like a human. I don't expect it to. I'm hoping that AI will prompt us to have better ideas, and that for that we need young minds. We need young people to be in front of that information so they can have innovative ideas.
Now, I mean, oh, sorry.
Well, I was just going to say, and we can't afford really to lose 40% of the world's talent and not have it participating on all the problems we have to solve. I don't think the world can afford to lose 40% of its talent because of bias, discrimination, lack of resources, poverty, other things. We really need to figure out access all the talent on Earth.
Yeah, that's a huge thing. And so many people are getting laid off right now. It's like, and a lot of people at the top are feeling like AI is going to solve all their problems. They don't need as many people, right? And there is the flip side optimistic view of like, we'll have more that we can do with AI. We'll have more problems that we can go after, more innovations, but it definitely doesn't seem like that's the direction where a lot of these companies want to head. It seems like slimmer is better for them. And I do agree. I think we've already had enough problems with K through 12 where we're not getting the maximum out of the human potential that we have, you know, and
Yes.
and like we talked about earlier, I think humans and what what our minds can do and what our bodies can do, et cetera, are just some of the most awesome transformers of chaos into order. That I say that human beings are more akin to DNA than they are to Homo erectus in the way that we store information and can then create creations out of the information that we store, in a sense, where obviously DNA creates organisms. We might create computers, or we might create airplanes, or we might create microscopes, or we might create et cetera, et cetera, et cetera. All these, higher order machinations of the way that we share information, store it and process it and create with our hands. It's really awesome. I mean, what
Yeah.
we've been able to do as a species, and to not see that and to reduce it to a bottom line, or reduce it to a financial output number, or reduce it to your worst, racial epithets or something like this, right? Like, it's just really unfortunate,
Right.
I think, going in that direction
I
and
mean,
away from the other direction.
humans have curiosity and imagination. And that means with curiosity, you have to ask questions. And imagination comes from who knows where. I mean, people jump up in the middle of the night and said, "Oh my gosh, I have a new idea." And where did that come from? And so I don't think AI can do that. AI doesn't ask questions. You have to prompt it. It gives a lot of answers, but the thing that really advances things is to ask the right question.
Yeah.
And so we need curiosity and then we need imagination. I'm just not sure that AI can do all that.
Now, yeah, I agree. I think you're in the majority maybe as well. I think a lot of people are starting to feel that way. Maybe not 2 years ago, but now it's like, okay, where's the money? Where's the is there an AI bubble? There's a lot of talk around that. And yeah, a lot of people saying like, where's the big outputs minus just it's really great though in a lot of different ways, but quantifying that or bringing actual value, it will be seen, right? And we definitely still need the humans in the loop, at least for now. I was thinking earlier about what you were saying, where every cell is programmed in that way to have all the information, right? Every cell has all the information. It reminded me of the Yamanaka factors and this ability to change skin cells back into stem cells and then transmit those into any kind of cell.
Right, exactly. Right.
That
Make
whole
it a
thing is,
brain cell. Yeah.
I mean, even just thinking about the possibilities of that. Oh, it looks like I just lost all my focus. So, any last words or last thoughts for young people or et cetera that you want to give before we wrap it up?
Well, for young people in particular, I hope that they can find or learn or experience the beauty of biology and that they can get in and take it deeper than my generation has, and that they can use their own curiosity and imagination to see things beyond what we know and can see now. That's really what science is developing tools or trying to understand the things that you can't see. And so it's really, in my opinion, you asked me this earlier, it's a faith exercise. Science is a faith exercise. It is the process of trying to find the substance of things hoped for and the evidence of things not seen. That's what science is trying to understand what you cannot see and then imagine how to see it or to understand it and to believe that it's there.
Wow. Wow. And that's precisely what you did in the facial recognition of the the spike protein, for example. And then figuring out exactly what those proteins look like and being able to save the world, really save millions and millions of lives. So I really appreciate you for coming by, and I'm really grateful for you coming here, but also, of course, for all the amazing work that you've done. And so thank you for coming by National Champion Radio.
Glad to be here with you.